Welcome to another lesson with Dr. Hill. Today we're in our biology series and we're going to be talking about genetic crosses, monohybrid crosses. And the best way to think about these things is to think about an example and work through examples. And so that's what we're going to do today. We're going to look at the first level, the first level cross of an example. So in this particular example, we're going to look at a gene for coat pattern. Okay. Now we've observed perhaps in some small rodent that they either have stripy coats or they have spotty coats, and the gene has been isolated, and it turns out that one version of the gene, if it's present and is dominant, will give a stripy coat, and the other version of the gene, the other allele of the gene, will give a spotty coat, but only in the absence of the dominant allele. Okay, so that's descriptive, and you quite often will get in a question in an exam that kind of description. And what you have to do is come up with a way of labeling that problem. And then what we do is we say, if we have a male who is heterozygous, and that mates with a homozygous stripy female, what is the outcome of that cross? What's the potential outcome of that cross? Say they have 10 offspring. What roughly are the offspring going to be? Okay, so let's have a look at that problem on paper where I've laid out the information, and we'll see how it looks. So over here, I have an example of the monohybrid cross. Remember, it's a gene for coat pattern. Well, I can't use a C for coat pattern because big C and little c look the same. Try and avoid these kind of letters. P is the same problem, big P, little p. So because it's a small rodent of some kind, I'm going to talk about hair pattern, and I'm just going to use H and little h. It doesn't matter what letter you choose. You're only talking about one gene, and it's important that you make it clear. So H is the dominant allele. It gives a stripy coat, and it's dominant. As long as it's present, the phenotype will be stripy. Little h gives a spotty coat and will only show when it's homozygous. Okay, so remember we were asked what happens if a heterozygous male for coat pattern gene crosses with a homozygous stripy female. So that means she has to be homozygous dominant. And there we go. So what we're going to do, so we've written it out. That's the heterozygous genotype. This is the homozygous dominant genotype. She could have been homozygous little h, but then she wouldn't be stripy, would she? The question said stripy, so she would be spotty, so she has to be dominant. Okay, so what we'll do is we'll do our cross, and if I take my Punnett Square, remember, noughts and crosses, and we'll put our female at the top. That's big H, and big H, little h for the male. So we've got our female up here. We've got our male here. Okay. And then what we do is we fill in an H from here and an H from here. One from here and one from here. One from here and one from here. And one from here and here. So what we would expect in this cross is 100. So we'll get 50%. Let's talk about the genotypes first. 50% H and 50% H, big H, little h. So we're 50% homozygous dominant and 50% heterozygous. What are the phenotypes though? The phenotypes will be 100% stripy. Okay, because they all have the dominant allele. So all of the offspring in this cross are going to be stripy, and we would expect that to be a pretty foolproof cross. If I cross those two individuals, I'll get all stripy individuals. Now, my problem is that I don't know, just by looking at the offspring, which of the individuals is heterozygous and which of the individuals is homozygous dominant. I know that this must be true from my original cross. But say I want to pick out, I want to find one of the heterozygous individuals. What can I do? Well, this is what we call the parental cross, P. Okay. Now what I can do is I can take my offspring and I can do another cross. So say I had 10 of these offspring, I've got 10 individuals here. I can expect 5 of them roughly to be that and 5 of them roughly to be this. But it might not work that way. Genetics is not determined, it's probability. So it's every possibility that it could be 6 heterozygous and 4 homozygous dominant. So in order to separate these guys out, I need to go to another cross. Say I'm a breeder of these animals and I want to, maybe they're pets or something, and I want to produce spotty ones for my pet shop and sell them to people who like spotty rodents. Then I have to find, I have to somehow figure out how to get myself some little h individuals. And they're hard to spot because the little h allele is in here, but none of my offspring are spotty. They are all stripy. So what do I do? Well, that's a problem that we're going to look at in our next video. So far, we have predicted the cross of a single, we predicted the outcome of a single monohybrid cross. If a heterozygous male crosses with a homozygous dominant female, all of the offspring are going to be stripy. They're all going to be showing the dominant phenotype, but half of them are going to be heterozygous and the other half are going to be homozygous dominant. All right, that's the answer to the first cross. The second part of this question, though, is how can I get myself some true breeding recessive individuals to stock my pet shop with spotty coats? Okay, and we'll talk about that in our next lesson. That problem is going to require us to do another 2 crosses. We're going to have to identify individuals, and then we're going to have to try and get ourselves some offspring that are homozygous recessive. So we'll see how that works next time. I'll see you in the next lesson.